In 2022, a DNA sequencing run in a Texas lab revealed ancient haplogroups X2, Q1A2, and an unknown autosomal cluster, all from a single 12,000-year-old limestone cave layer. The results didn't just challenge the accepted migration timeline, they broke it. If the single origin story was wrong, how did these distinct genetic signatures end up side by side? And what does that mean for everything we thought we knew about how humans first arrived in the Americas? The limestone chamber in central Texas was sealed for over 12 millennia, its entrance hidden beneath layers of sediment and caliche. When field crews first uncovered the burial context, every step was logged, every tool sterilized. Inside the lab, the team followed strict contamination protocols. Negative controls, so-called blanks, were sequenced alongside each sample, tracking any trace of modern DNA. The extracted material yielded an average sequencing depth of 1.2 times, a figure that signaled both the age and the integrity of the find. The genetic profiles came from the same stratigraphic horizon, a layer undisturbed since the Pleistocene. Each sample passed authentication checks, cytosine deamination at fragment ends, short average molecule length, and a damage pattern consistent with ancient preservation. The lab's internal audit flagged nothing unusual in the workflow. Every reagent batch was cleared as DNA-free. The sequencing run produced 3 distinct haplogroups, each confirmed by independent rounds of library preparation. The team repeated the process using a second set of bone fragments. The results matched Frost. The evidence was locked in stone and sequence, impossible to dismiss as error or contamination. Textbook models predicted a single founding population for the Americas, one wave crossing Beringia, then fanning southward in isolation. The expectation was clear. Early remains from the same time and place should share a uniform genetic profile, with minor variation at most. When the Texas samples returned, 3 sharply separated haplogroups, the reaction was immediate. The numbers didn't just drift, they split, with bootstrap support above 99% for each lineage. Admixture graphs based on F statistics showed deep divergence, not a single stem. The genetic data, once cross-checked, stood firm. The lab's population structure analysis mapped the Texas haplogroups against reference panels from across the Americas and Northeast Asia. Instead of clustering tightly, the samples fell into separate branches, one matching the expected Beringian ancestry, the second with markers more common along the ancient Pacific Rim, and a third showing affinities that didn't fit any known migration. These were not minor deviations. The F4 and D statistics produced Z-scores well above conventional cutoffs, confirming the lineages were distinct. The team ran contamination checks and statistical bootstraps, but the signal held. The Texas burial layer contained genetic diversity that the single migration model could not explain. For decades, the prevailing theory had treated population structure as a later development. Here, in the heart of the continent, the evidence pointed to multiple ancestral streams arriving far earlier than expected. The contradiction was not theoretical, but written directly into the DNA. Kern stood over the sequencing results, eyes tracing the branching lines of a beast phylogeny chart. The mitochondrial DNA extracted from the Texas remains carried markers found today along the Pacific Rim, haplogroups that trace their deepest ancestry not to the Siberian interior, but to populations clustered along ancient shorelines. Y chromosome analysis added another layer. The dominant lineage matched those common among maritime groups in East Asia, not the expected Beringian signature. Autosomal dating pushed the earliest divergence back over 18,000 years, predating Clovis by at least 5 millennia. That figure was not a statistical fluke. The confidence intervals remained tight, even after cross-checks against reference panels from both the interior and the coast. The genetic evidence pointed to a migration event that began along the Pacific Rim, then pushed inland, deep into the continent, more than 1,000 miles from the nearest ancient shoreline. The Texas cave, far from any coast, preserved genetic signals that mapped to a maritime route. Kern highlighted the SNP M120, a variant tied to seafaring populations, present in the Y chromosome data. The implication was clear. The first arrivals were not Ice Age hunters following herds through an inland corridor, but people adapted to life along the water, whose descendants reached deep into the continent long before the ice-free path opened. Limestone caves in central Texas act as natural vaults for ancient DNA, preserving genetic material where most North American sites yield nothing but dust. The chemistry here is precise. pH readings hold steady around 7.2, neutral enough to block the acidic decay that destroys bone elsewhere. Calcite crystals grow along the chamber walls and within the bone itself, locking DNA fragments in place through a process called adsorption. This mineral shield prevents water and microbes from breaking down the genetic code. Average fragment lengths from the site reach 85 base pairs, short but intact enough for full mitochondrial and partial nuclear sequencing. In contrast, bones from open-air sites in the Midwest or Northeast rarely yield fragments longer than 30 base pairs, if any survive at all. Relative humidity inside the cave remains below 60% year-round. The result is a genetic record as reliable as any recovered from Arctic permafrost, but found in the heart of Texas. This preservation window is rare, and it is the reason the data emerging from these caves is so robust, unfiltered by the usual noise of contamination or decay. Texas genetic data, when run through QP-Graph and compared to every known alternative, fit best with a three-wave scenario. The proportions were clear. About 40% of the ancestry traced to an early Pacific coastal migration, 35% to the classic interior Beringian route, and 25% to a later, more complex influx. The numbers weren't guesswork. They came straight from the data. The model's fit wasn't just better, it was decisive. The delta AIC value, a measure of statistical support, dropped by 212 points compared to the old single-wave model. That kind of gap leaves no room for ambiguity. No single origin or two-wave model could account for the observed diversity. The three-wave synthesis aligned with the full spread of haplotypes, admixture signals, and temporal overlaps found in the Texas samples. The evidence forced a new view, one where the continent was peopled by multiple streams, converging and mixing deep in the interior. The model didn't just fit the Texas data, it demanded a re-examination of every early site across the Americas. The numbers, the sequences, and the statistics all converged on the same point. The old migration story no longer held up. Genetic data rewrites the map in real time. As ancient DNA continues to surface, every new strand challenges our oldest assumptions. Migration isn't a closed story, it's an evolving equation. In science, the evidence leads. And tonight, it points to more questions than answers. What do you think?